All right. The first one says foods are digested before they are absorbed into the blood. Yes. Figure one shows organ in the human digestive system. Okay. So yeah. Well, it says which organ is the stomach. Okay. So uh, we are pretty sure of this one. The organ mark, the letter that points to the stomach is C. So that should be uh, the correct answer for this one. Um, okay. So now the second one says what type of enzyme is produced in the stomach? Okay. Now that's one I'm not too sure, but I can take a shot at it. Um, I I'm looking at now. Is that the only? Are these three the only options, or there's a fourth one that we can't see? Okay, that's
No,
all. Okay, I can see. That's just three of them. Okay. Um, I'll go with lipase. I'm not sure of it, but we can check with the marking scheme just to be sure. What does it say?
it says protease.
Protease. Okay. Okay. And protease looks like it helps to break down um okay, I mean not but protease seems like it's adapted to protein, while lipase seems to be adapted to fat and fatty and fats and yeah, uh, fatty food. Then carbohydrate should be for carbohydrate. Okay. All right. Let's move on. Okay. It says which term describes the pH in the stomach. Give one reason why the stomach is this pH. Okay. So hmm, ah, the the stomach should be uh okay. So I think stomach is uh alkaline. Okay. Okay. So the question now is to give the reason. Okay. Uh, okay. Okay. So I'm not too sure of the reason now, but so food are usually broken down. Uh, um, have fatty food broken down into fatty acids. Okay. So I'm even thinking whether it might be acidic just because of the reasons that fatty foods will be broken down into fatty acids, become fatty acids. Uh, so I might go with acidic based on that reason. Okay. But what does the market thing say?
Yes, it says acidic.
Okay. Does it give a reason why?
produces hydrochloric acids, which allows um best condition to digest food and proteins.
Okay, okay. Food and protein. Okay, okay, okay. All right. Okay. Let's go to the next one. Which organ produces bile? Um, bile is that I think it's produced by the liver. It's not the mouth. I'm sure it's not the mouth. I'm equally sure it's not the large intestine. So it will be between the liver and the pancreas. But I want to go for the liver. Is that correct?
Yes. Um, what is bile?
What did you say?
What what is bile?
Okay, it's also one of the juices that aids in digestion. So um well let me see if I'm trying to remember um um its use now. It breaks down, it helps with digestion of let's see. But let me see. So um it aids digestion generally. Okay, so I think it's not basically for any um class of foods. It's just for foods, general classes of food, but it aids digestion and it's produced by the liver. Then if there's a question about where it is stored, that's the gallbladder. Uh, yeah. Okay. So we can move on now. Okay. It says, how does bile help in the digestion of food? Okay. Uh, okay. So now I'm trying to see which one is correct. Bile is alkaline in nature, so that last option cannot be correct. That it makes the pH in the small intestine acidic. It's not possible. So that last one is out of the picture. It increases the surface area of fat. I don't want to go with that. Um, but the middle one looks more like it. Or no, wait a minute. It's an enzyme that digests protein. So is the bar really an enzyme? It's a liquid, a fluid. Uh, I can say it contains something, but then not that it's an enzyme itself. Um, so I think that first one that says it increases the surface area of fat by breaking it down. Yes, I think I'll go with that. That first one should be correct. It increases the surface area of fat. Typically, fats are a bit difficult to digest, but bile somehow helps to break them down. So, yeah. So it is not an enzyme itself, but if we want to say that it contains enzymes, that's a given. But um, that first one should be the right thing. Okay, so just for our just because we have seen it before, you remember that earlier the question was asked that what is the nature of the pH in the stomach, right? And we saw that the stomach is acidic.
Yes.
So the body, you know, the body's environment to be balanced, right? We can't have a an always acidic
Yes.
environment. So the bile, being alkaline in nature, helps to balance that the acidity that is in the stomach. So that's the way the body naturally helps itself. Um, um, by the bile. So the bile doesn't, it's not produced by the stomach. Hello, are you there? So I think your mic is muted. Okay, I can see your screen, but your mic is mute. Okay. Um. Okay, I can. Yeah, I think it's unmuted now. Okay, but your screen, I can't see your the question paper anymore. Okay, it's back now. All right. Okay. So, okay, so you got that extra info about the bile, right?
Yes.
Okay. All right. So it says a student tested different foods for the presence of protein, starch, and sugar. It says draw one line from each food molecule to the reagent used to test for the food molecule. So I know iodine solution is used to test for starch. Uh, I'm not too sure of the other one, but let me see. Benedict solution. Iodine is for testing of starch. Um, uh, Benedict solution should be for uh, sugar. Yeah, it should be for sugar. Okay, so that will now leave us with biuret for protein. So, but I'm not sure of that last one. So Benedict solution should be for sugar, while iodine is for starch. So just confirm it that the last one is biuret for protein.
Um, Bury it vegan.
Biuret is for what?
Protein.
For proteins. Okay, so that means we're good. Okay, all right. Okay. So it says give one safety precaution a student should take when using Benedict solution. Okay. One safety precaution. Um, Okay. Let me see. Um, so maybe we should uh, talk about a little about what happens when one is using the Benedict solution. Um, the basic thing is the skin. I think the skin, basically, it could affect the skin, and I think that's a given because we said Benedict solution um is for sugar, for testing reducing sugar, which is sugar technically. Okay, starch is more like a complex sugar, so that's why iodine will be suited for it. So um, Benedict solution is for basically sugars, and those things are, you know, somehow um, uh, would the solution itself is um, could cause irritation to the skin because of that its nature. So irritation to the skin. Um, well, typical experiments, you protect your skin by wearing, gloves and stuff like that. Then you may also think about googles for the eyes. So yeah, so so if we I know I'm sure about the skin, then we could also add um wear googles for the eye. So that should be a safety precaution. Okay, okay. So does the marking scheme give anything extra in terms of Benedict solution precaution?
Wear gloves, wear goggles.
Okay, 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 perfect. Okay, all right. Table one shows the results for one food sample. Okay, Benedict test is red, virus test is blue, iodine test is black. Okay, it says which of the tests show positive results? Okay, so which of the tests shows positive results? Um, let's see. Red, blue. So I'll go with iodine test because iodine test the presence of starch in 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 a sample, right? If iodine touches that sample, it should turn from white to blue black. So I'll go with iodine. Okay, but now it's saying it doesn't give us iodine only. So it's going to be one of these, either all three tests or Benedict and iodine only, or virus and iodine only. So we need to be sure. Okay, okay. So okay, I'll go with Benedict test and iodine test, but I'm not sure of virus test. Turning blue. Okay. Okay. And virus test is for iodine is for starch. Benedict is for reducing sugar. And virus is for protein. Okay. Okay. If we if we want to, I think I'll go with that one. But let's just confirm with the marking scheme. I'll go with Benedict and iodine test only. I'm not sure, but Benedict and iodine, those ones I'm sure. Oh, what does the scheme say?
Benedict's and iodine testing.
Okay, Benedict and iodine, right? Okay, okay, okay. So I think that virus is not a positive test. Okay, all right. So we'll go on. The next question says start molecules are not absorbed into the blood from the digestive system. Give one reason why. Well, it's quite obvious. Starch is a complex sugar. It's the blood will only be able to absorb glucose, the simplest form of sugar. Starch is insoluble in blood, to not be able to dissolve in blood and be absorbed. So it has to be broken down into simpler sugars. That's the reason why it cannot be absorbed into the blood from the digestive system. Okay, but let's see if the marking scheme gives us another reason. Do you want to check?
Okay. It says starch molecules are too large.
Mm,
Starch molecules are insoluble.
okay, okay, that's fine. So I think we pretty much covered it. Okay, so let's move on. Figure two section through a leaf. Okay, okay. It now says, give one way that the palisade layer is adapted for photosynthesis. Okay, the palisade layer. Let's look at this thing properly. A cross section through a leaf. Uh, okay. So if you look at the nature of that cell, the cells there, that in that palisade layer, they look different in their structure from the others. Uh, if you look at them in terms of their size, they have the advantage of having more of chlorophyll, just based on their size, and then the way they look like pups, more like you know, and they will be able to basically function well, when it comes to converting light energy um, into the energy that's useful for photosynthesis. So yeah, the their location too directly not exactly directly under the but they are directly under the topmost layer of the leaf. So yes, they um they are well suited for photosynthesis um, in that aspect. So we can say their shape. So you can talk about their shape. They are they are looking cylindrical, or the way they look. they look more like cylinders, not like the regular spherical shape that other cells have. And then, and the fact that they have, they contain chloroplast that help to convert the uh, light energy to chemical energy for photosynthesis. That would be how they are well adapted for photosynthesis. Okay, okay. So does the market scheme say more? Hello, Noah.
Yes.
The market say more about the palisade layer.
Pardon?
Does it say more about the palisade layer in the leaf? Why, how it is adapted for photosynthesis? The marking scheme.
Um, allows chlorophyll for chloroplast.
Okay.
Cells contain many chloroplasts. No gaps between cells. Cells
Mm-hmm.
are clear.
Okay. Okay. So yeah. So its adaptation. Okay. Can make it well adapted for photosynthesis. Okay. The next one says gases pass into and out of the leaf through small pores in the surface of the leaf. What are the small pores labeled X called? Okay, we know those are called the stomata. That's uh, the name. So the stomata is the correct name. Okay, so we can move on. Okay, it says a student viewed a section of a leaf using a microscope. The student measured the length of one of the palisade cells. The cell image measured 28 millimeters in length when viewed at a magnification of times 400. Okay. Calculate the real length of the palisade cell in millimeters. Alright, it says use the equation real length equals image length over magnification. So, I mean, we have a image length that's 28. 28 divided by 400. Okay, so think about it. 28 divided by 400. 4, you can use 4 to break it down, right?
Yes, yes.
So what would you have on top? 4 into 28. That's 7, isn't it? Right?
Yes.
Okay. Then 4 into 400, that's 100. So if you say 7 over 100, what would it be? What is the answer in decimal?
0.0.
Over 100. Good. 0.07. Yeah. So the decimal point will move to the left 2 times. So that's 0.07. Okay. Okay. So yeah, let's move on now. 0.07 millimeters. Okay. It says convert the real length to of the cell from millimeters to micromillimeters, and 1 millimeter is equal to 1,000 micromillimeters. Okay. So when you're converting from millimeters, remember what we talked about yesterday? Conversion measures, right?
Yes.
Do you remember? Talks about 100 centimeters make 1 meter, so you know whether you are multiplying or dividing. In this case, we want to convert from millimeters to micromillimeters, and that's going to be um, um, in this
Um,
case, we're going to be multiplying by 1,000, isn't it?
yes.
So now remember, when you multiply by powers of 10, the decimal point will move to the right hand side. So we're dealing with 0.07. Multiply 0.07 by 1,000, what will you get?
70.
70 micrometers. Okay, so let's not lose sight of that fact. That's 70 micrometers. Okay, so let's move on. Carbon dioxide. What? Carbon dioxide can move in and out of cells. It says, what is the process by which carbon dioxide can move into and out of cells? Carbon dioxide. It's okay. So if I go with the fact that it's a what do you call it? It's a gas. I think we would use go. I'll go with diffusion. But what does the marking scheme say?
Um,
Is it correct?
yes, diffusion.
So remember, diffusion is the process of a gas, usually a gas, moving from an area of higher concentration to an area of lower concentration. You can easily remember examples like spraying a perfume, you know, from a point, at the point where the perfume is sprayed. That's the highest concentration it can get. And then by the time it diffuses to other parts of the room, it will get to further parts of the room where the concentration was low. So that's the diffusion. Okay, so let's move on. While osmosis seems to be like the opposite of diffusion, from an area of lower concentration to an area of higher concentration through a semi-permeable membrane. That's the difference between osmosis and diffusion. Okay, so you got that?
Yes.
Okay, all right, let's move on. All right, it says figure 3 shows a diagram of four cells: cell A, cell B, cell C, and cell D. Uh, those dots represent carbon dioxide molecule. What does it say? It says which cell will carbon dioxide move into at the fastest rate? It's obvious. Carbon dioxide will move into a place that has more space for it. What do you think? Isn't it?
Um,
Okay. So which one do you think um, um, carbon dioxide will move into? I'm looking. The answer would be between cell A and cell B. But can you notice that cell A has more carbon dioxide around it than cell B? But the carbon dioxide inside is uh, is the same in terms of the level. Okay, so I'm saying that the fusion will happen faster in cell A. What do you think?
um, sorry, can you say why you think it's A again, please?
Okay, I feel it's A because number one, the concentration of carbon dioxide around cell A is high, remember what diffusion is. Movement of like molecules from an area of higher concentration to one of lower concentration. And then the cell A2 has more space for the carbon dioxide to occupy. It's not like cell C or cell D that seem to be already filled up. Do you get?
Yes.
So that's my reason for choosing A. Okay, but I was just asking, do you see reason with me, or do you have a different opinion?
No, no, no. I just didn't understand it at first. But
Oh, okay, okay, okay. But what does the marking scheme say in terms of the reason?
um steeper, so steeper diffusion gradient, bigger difference in concentration of carbon dioxide inside and outside the cell, high
Okay,
concentration of dioxide.
okay, okay. So it agrees with us in terms of A, right?
Yes.
Okay, perfect. Okay, all right. So that's it. The difference in the concentration, the obvious difference, the high difference, and then you know, yeah. Okay, of course, an experiment will follow. Okay, so what are they really interested in? Um, what two measurements should the students take to calculate the rate of photosynthesis? Obviously, is the height of the gas. The height of the gas in the that's coming out from the conduit. So you can see those bubbles of gas there, right? Those bubbles are showing that um, the conduit is producing oxygen, you know, to as it's coming out. The oxygen, excuse me, the oxygen is coming out, and as photosynthesis continues happening, more oxygen will be coming out, right? And what will happen? The that space there will be increasing and thereby push down the liquid. So for him to be able to measure the rate of photosynthesis, he will measure the time taken, and the height of the gas column, because that column is going to keep on increasing as more gas is produced. Did you get that?
Yes.
Okay. Okay. All right. So we can move on, right? It says give two variables the student should keep the same in the investigation. Okay. Two variables the student should keep the same. So that they don't influence the kind of control variables. Um, hmm. I think the volume of the liquid that the conduit is in. Is that should be water, right? It should be water, isn't
Yes.
it?
Yes.
Okay. So the volume of water used should be fixed. Let me see what else should we keep constant. Um, okay. I am not too sure now, but that one is sure. The volume of water used is- should be kept constant. But what does the marking scheme say about the second one?
Um, temperature of water,
The
light
temperature,
intensity,
light intensity.
Distance of light
Okay.
from pondweed.
Okay, the distance of the light from the conduit. Okay. Light intensity, distance of light from the conduit, and temperature of the water. Okay, perfect. Okay, so let's move on. Um, okay, table two shows the result. Blue, green light, and red light. Rate of photosynthesis 9, 1, and 8. Okay, so what's the question? It says,
Uh,
it says you show a okay. So if you were to draw this graph, what do you think will be the x-axis, and what do you think should be the y-axis?
colors would be the x-axis, and the rate of photosynthesis would be the y-axis.
Perfect. Yeah, I agree with you because a bar chart needs something that will show different levels of rises in that will help us to show the different heights of bars, and that only can be afforded us by the rate of photosynthesis, which is a number. So the x-axis will be the colors of the light. Perfect. All right. So let's move on. Okay. So somehow they even gave us the what the x-axis should be and what we should label the y-axis. Okay. No problem. Let's move on. Table two. It says what color of light should be used to grow plants in a greenhouse? What color of light? Okay. So I think it's the one that will give us highest photosynthesis, isn't it? What do you think? Noah. Hello, Noah, are you there? Hello. Hello, Noah, you there? Mm. Hello, Noah, I can't hear you anymore.
Sure.
Okay, Noah,
Yes, hello.
what happened? Your internet connection?
Yes.
Okay, all right. Okay, you can go ahead and share your screen again.
Sorry, just let me screen record.
Hello, Noah, are you there? Okay, all right. Your mic is muted. I didn't hear you. Okay. So I think the last question that we um okay, submitted now. The last question that we were working on was on the one that asked us which light should be used in the greenhouse. So we said, I think we agreed that it should be the blue light, right? Hello, no, are you there?
Hello.
Okay, I can hear you. You can go ahead and share your screen.
Thank you. So yes, blue lights.
Okay, all right. So yeah, we'll move on then. Okay, it says this question is about disease, road, black spot is a disease where black spots develop on the leaves of rose plants. What type of pathogen causes rose black spot disease? Okay, to be very, 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 very frank, um, let's see. Is it a bacterium? Is it a fungus? Is it a protein? Or is it a virus? Okay, so I'm trying to see because sometimes um certain classes of diseases and certain diseases produced by either a virus or a fungus or a bacterium have a certain way they look or have a certain way they attack plants. So you can use your knowledge based on that to tell, you know, um, which one it is. Uh, so for this one, um, let's see.
Is it is it fungus?
Is it okay? Does the market scheme say it's a fungus?
Um, yes, it says fungus.
Okay, okay, okay. It says plants with rose black spot disease often have yellow leaves. Suggest one reason why the leaves are yellow instead of green. Okay, so I mean, from what causes the leaf to be green is that uh, the fungus will attack most likely attack the um um what gives the leaf its color. Uh, that would be uh, Okay. I think it's the chlorophyll.
As in they have less chlorophyll, or
Less, yes, less. Yes, I think that's why. Yes.
okay.
What does the marking scheme say?
Um, no chlorophyll, or the chlorophyll has been broken down.
Okay, okay, all right, okay, that's fine. That's fine. And the chlorophyll is what responsible for the leaf green color. Okay, all right. So let's see. The next one says explain why plants with yellow leaves will grow slowly. Well, I mean, they definitely. It's not as if there's no photosynthesis happening, but of course, the rate of photosynthesis will be affected because the leaves are weak, uh, and and not as efficient as they would have been if they were healthy. So the low chlorophyll would affect rate of photosynthesis. So yeah, that so that would lead to stunted growth, right? It doesn't it doesn't stop photosynthesis entirely, but it slows it down. So that would lead to stunted growth. Okay. Does the marking scheme say something different?
Less photosynthesis or light being absorbed.
Mm, Okay. Okay. Yeah. Okay. So we're on track. Okay, let's move on. Okay. It says um oh all right. Well, draw one line from each method to the control of control to the explanation of how it works. Okay. Remove and burn infected leaves. Okay. So it says you should draw one line from each method to the control of control to the explanation of how it works. So if one burns infected leaves, what does that help to do? I think it creates um it's uh it creates a barrier to the moving of the pathogen. Um, okay, no, I'll go with it reduces the chance of the pathogens being spread by water droplets when you remove them. Um, yes, yes, when you remove them, it reduces the chance of pathogens being spread by water droplets. Uh, the second one says water the roots of the plant only, not the leaves. Oh, okay, okay, okay. I think that one is more suited to water the roots of the plant, not the leaves. Yes, it reduces the chance of the pathogens being spread by water droplets. Then remove and burn infected leaves. Pathogens are killed. Yes, those are the two I'll go with. What does the marking scheme say?
The first one is the pathogens are killed.
Oh, excellent. Okay, second one.
It's called reduces the chance of pathogens being spread by water droplets.
Okay, perfect. Perfect. All right. So did you see? I think sometimes one needs to just critically look at, the options. But yeah, sometimes, just looking at the options will give you a chance of making sure that the answer you chose was correct. Okay, let's move on. Uh, It says tobacco plants may become infected with a pathogen called TMV. What type of pathogen is TMV? It sounds like a virus to me. Not too sure of TMV, but it sounds like a virus to me. Tobacco, tobacco. Yeah, yeah. What does the marking scheme say?
Yes, it's a virus.
Okay, okay, okay. So it says next for malaria. It's a disease caused by a protist. How is malaria pathogen transferred to humans? An infected, even though then I'm not sure if research has changed it, but then we were told that it was caused by an infected female mosquito carrying plasmodium. Okay, so we want to scroll down. Are there options? Okay, how can this? Oh, okay. Okay, it says how is it? So we're supposed to explain. So basically, what I know, you can confirm from the marking scheme, is that it's spread by being humans being bitten by um, an infected female mosquito. Okay, that's carrying infected in the sense that it's carrying plasmodium, which is um, the protein that causes malaria. Okay, so that's for the explanation how. Then the next question, how can the spread of malaria pathogens be reduced? Uh, it can't be reduced by avoiding sexual contact or cooking food thoroughly. Draining water from swamps sounds more like it. Yes, that's the answer. Because the mosquitoes usually breed that way. They breed by um, through water. So wherever there is water around, a female mosquito and a male mosquito would find that a very good ground to lay their, the female would find it a good ground to lay their eggs and start the process of the growth for a mosquito, for the next generation of mosquitoes. Okay, so yeah, that's how to control the spread of malaria pathogens. So one may be wondering, if an infected mosquito bites a human, right? And that human now gets malaria. Why should breeding more mosquitoes be the gateway to um, spread of malaria? Because what happens is that that person who now has malaria, right, will get could get bitten by another mosquito, by another female mosquito that's not infected. Guess what? Through that bite, that mosquito now gets infected and can now bite another person that doesn't have malaria, and that person will now get infected too with malaria. So that's why draining water would help to curb the spread of malaria pathogens because they will not have anywhere to breed. Mosquitoes usually like breeding with open spaces where water is, like containers and swamps. Okay, so I think we can move on. Or do you have any questions about that?
No, no, no.
All right, I'll move on then. Okay, okay, please turn over for the next question. All right, cigarette, cigarette smoking is the main cause of cancer in the UK. Mutations in cells cause cancer. Where in a cell do mutations happen? Hmm, I would go for the nucleus, and that's the reason why it's really dangerous. So, does the marking scheme agree with that?
Yes, it's the nucleus.
Okay, all right. It says, why do some cancers develop into life tumors? Because of growth, period, nothing else. So the first option is correct. Cells never stop dividing. It's growth, nothing else. Okay, we can move on. Unless you want to check the marking scheme for um, to be sure, but I'm sure of those two. Cigarette smoking has been linked to many different, yeah, obviously. Lung cancer is the most common type of cancer caused by smoking. So that's one reason why. Hmm. Hmm. Well, if you were to suggest a reason why, what would you say? What would you say?
You're inhaling chemicals.
Yes, but not just that. The fact that smoking, right? How is cigarette usually smoked? It's inhaled, right? Usually they inhale deeply, right? And when they inhale deeply, guess what happens? The smoke goes into the lung. Smoke is not meant for the lung. So that smoke with all the nicotine, with all the dangerous chemicals, the first point of contact with the body, apart from the mouth, of course, and well, of course, okay, when it's coming out through the nose space, is the lungs. So, I mean, it's obvious why uh, that's those are the cells that would most likely be affected. But what does the marking scheme say something else? I guess they would have something more detailed. But that's what came to my mind.
Chemicals are enter the lungs. Chemicals are inhaled. Chemicals are more concentrated in the lung.
Exactly. Exactly. Okay. So we move on. Describe how this can happen. That second one is basically what happens now. I mean, body organs are tightly packed. So as one is, as uh, they spread by cell division. So cell division, one cell um, um, affects the other. And imagine that blood is still flowing through those infected cells. So that's the way it will spread. I mean, uh, 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 both by contact and through the blood. It's very it's very easy for it to spread. But what does the marking scheme say about spread? Secondary cancer.
Cancer cells are transported in the blood.
Mm-hmm. 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 Okay, they're transported in the blood. Okay. Okay. So that's the primary thing. Okay, let's move on. It says sometimes a person may need a lung transplant. What does it say? The NHS will not offer one to a person who smokes. Why? Mm. Okay. I'm not too sure about this, and I'm not too sure why. Could it be because um it's a habit, right? Most likely, if such a person gets one, the person would probably relapse into the same habit. I'm not too sure why, but that's what I just think. But what does the marking scheme say? Does the marking scheme say something else?
Sorry. Smoking will damage the new lung, limited finance.
Mm-hmm. Mm-hmm.
The lungs could be used
Mm-hmm.
on else. Illness is self-inflicted or person is not attempting to help themselves.
Okay. Mm. So that's the problem. The habit thing is just out of this world. That's why sometimes um, breaking away from such harmful habits is is usually a challenge. It's better not to even start it than you know um start thinking of how to stop it. Okay, let's move on. Okay, okay, okay. This is a graph about skin cancer and all that. So what does it say? It says how many more new cases of skin cancer are there in males aged 40 to 44 than in males aged 15 to 19. Okay, let's look at the graph very well. 40 to 44, 15 to 19. 40 to 44 is that's 320. Yeah, if each box is 20, right? I'll take it as 320. And then 15 to 19 is just 20. So that is 300. 320 minus 20. Do you agree with that?
Yes. 16.
All right. Yeah. Okay. All right. So let's move on. It says there are no new cases of skin cancer diagnosed in males younger than 15 years of age. So just one reason why. Younger than 15 years of age. So that means they are basically younger teenagers, right? So could it be what are the primary things that cause skin cancer? Exposure to ultraviolet rays, um, use of chemicals, things for skin, skin chemicals that could be harmful to the skin. Those are the two primary causes I know. There could be more, but those are the two primary causes I know. Well, if you look at it technically, I think younger teenagers haven't really most likely are not trying to impress anybody. So most of them don't use um a lot of skin chemicals, right? Um then equally, I'm just equally looking at the flip side too. Um who are the most likely people you would see on a beach, right? Being exposed to um the sun, like most likely harmful rays from the sun. You would most likely find older people, right? And um, older teenagers, older people, and then the older they get, of course, that means that they go to work, they go to work, and that they are more exposed to sunlight, and by extension, harmful radiation from the sun, ultraviolet rays from the sun. So yes, um, um, teenagers who are usually younger than that may not have those chances, right, of being exposed to new cases of skin cancer. But that's just my own opinion. I don't know if the marking scheme has something quite different.
Um, little exposure.
Yeah, little exposure. That was one of the things I said. Little exposure to the sunlight or harmful radiation from the sun. What next?
More use of sunscreen.
Okay, 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 okay. More use of sunscreen. So that's for the younger teenagers, right? And younger children.
Um,
Yeah. Okay, that's cool. Sunscreen, yeah. Okay, I didn't. Yeah. So I talked about use, and that was even the in places like Africa, use of creams that are harmful to the skin. So but sunscreen are usually used by younger children. That's true. So that would prevent them from exposure to UV rays from the sun, which is a good one. Okay. All right. So what does it say? Give one conclusion from the data in figure 6. Well, what conclusion can we give? People in their prime. I think the graph, can you scroll up? Let's look at the graph one more time. I think people in their prime it seems to be higher in people in their prime who are more exposed. And that is 65 to 69 by virtue of the fact that they are working and they are outside most of the time. Yes. They're outside most of the time. They will rather use the sunscreen for their children than for themselves. So, yeah, especially when you talk about men are not really keen on, oh, take care of your skincare and all that. They're not really big on it as much as women are. So, yes, that's what I would say. One conclusion, yes, mostly males in their prime are the ones who have the highest incidences of new uh, skin cancer cases. And then you will see that it reduces as they grow older because obviously they will be going out less. So they are not really as exposed to the sun as before. So I think I don't know if that can be considered as one conclusion, but yeah. So what does the marking scheme say for that guy before we move on?
New cases each year are in males, people aged 60 to 60 to 64.
Mm, mm,
Cases per year increase from age
mhm.
15 up to 69.
And then they drop from 65 to 90, right?
Yes, yes.
Okay. Okay. All right. Is that all?
Yes.
Okay. All right. Okay. We move on. Survival for all types of cancers have improved over the last 20 years. I think we've seen a question like this. Suggest two reasons why. Well, obviously, improved um, patient knowledge, improved knowledge uh, of
So,
prevention, yeah, and what else? Better care, right? As
yes.
in research. So, yeah. Okay. So I think we can. Okay. Okay, this one is um about bacteria. Okay, so it says what are two similarities between a bacterial cell and an animal cell? Tick two boxes. Oh, nah, that's fine. Usually they say tick only one box. So let's see what similarity. Okay, so I think I'll go with the first one. Both have a cell membrane. But let's see what the second one should be. Okay, cell membrane, not cell wall. Okay, so I'll go with both have a cell membrane and both have cytoplasm. At least those are the two things I want to show in cells. So yes, the cytoplasm will be for the other things that are inside the cell to exist and function. So yes, I'll go with those two. Okay. So are we correct on those two?
Um, yes.
Okay. All right. Okay. Salmonella food poisoning is caused by bacteria in food. Says, why do the ones that are very, very obvious, they say we should not refer to them. Salmonella. Okay, I'm not too sure of this one, but because the obvious one is vomiting and diarrhea. I mean, I'm not too sure of other symptoms of salmonella. What does the marking scheme say?
Stomach cramps,
Stomach cramps. Okay, what else?
a fever,
Okay,
high
fever. Oh, okay. What else?
and sweating slash chills.
Sweating.
Yes, sweating from high temperature or chills.
Sweating from. Oh, okay, okay. Sweating. Okay. Obviously, because of the fever. Okay. So sweating and then from high temperature caused by the fever and stomach cramps. Okay. Okay. That's fine. Okay. Let's move on. Question five continues. We're still talking about bacteria. It says, what's the name of the first antibiotic developed? The name of the first antibiotic. Okay. Ah, I'm not sure about this. Okay. I'm not too sure of this. Uh, so I know. Okay. So there's this the person who helped to discover a way to preserve milk using the method called pasteurization. I'm not too sure now if that was the very first, but uh, antibiotics usually that one is not for. So that one is for like preserving stuff. We're talking about now antibiotics for the human body, right?
Yes.
Okay, okay. Um, hmm, penicillin. Would that be it? Okay, I'm not sure now. But what does the marking scheme say?
Um, it says penicillin. Penicillin.
Okay, okay, all right. That was a close one. I was not too sure, but yeah, that just came up. Okay, it says a child with severe bacterial infection was given a course of antibiotics. Figure 7 shows the concentration of live bacteria in the child's body change when taking the course of antibiotics. Okay, let's scroll down and take a look at the graph. Could you scroll up a bit? Let's see the graph. Okay, so apparently, as
Oh,
the
sorry.
course of antibiotics, you can it reduces, right?
Yes.
So over time it will. So okay, so what's the question that they're asking? Let's see the questions on there. It says the concentration of the live bacteria in the body continued to increase after starting the course of antibiotics. So just one reason why. Okay. Well, I think it's probably because the amount of live bacteria uh, uh, could not be contained by the first initial course of the antibiotic, right? And the body was probably trying to adapt to it. So, so it seems like the concentration of bacteria was increasing until a point where the course of the antibiotics increased sufficiently enough to be able to contain the concentration of the live bacteria. Okay, did any of what I just said now make sense?
Um, Not really, no.
Okay, so you understand that the bacteria all this while was having a field day, right, in the body. No contention, nothing was fighting it, right? So the course of antibiotics now got introduced. They will advise that you don't, the child doesn't take beyond a certain dose. So the dosage will increase in the body over time. Do you understand what I'm trying to say now? The dosage of the
Yeah.
antibiotics will increase in the body. So if you can't, it's not, it's if one wanted to just imagine if the amount of antibiotics taken after 5 days, the person now decide to take it at once, that would be overdose, and that could lead to complications. So it has to be taken in small quantities, and as they keep going. So in those small quantities, the initial small quantities of antibiotics could not stop the increase of the bacteria in the child's body initially. But as time progressed, you understand, it was able to contain it as as time progressed. But I don't know. Does it make sense now?
Yes.
Okay. But let's see, does the marking scheme say anything more?
Only a few bacteria killed, so live bacteria
Mm-hmm.
continued to reproduce.
Uh-huh.
Time delay for antibiotic reached bacteria. Time delay before antibiotic could kill bacteria.
Okay. 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 So let's see. What does it say? It says after 3 days of taking the antibiotics, the child felt better. There was still bacteria in the child's body. Why did the child feel better? Number one, it says the bacteria had become immune to the antibiotic. No. The child had become resistant to the bacteria. Yes. I'll, I'll say, I'll go with that second one. The child had developed some form of resistance to the bacteria. Yes. Is that the one the marking scheme said was correct?
Yes.
Okay. Suggest why doctors do not give antibiotics to patients with minor infections. Well, um, what I know about this, the little I know is that um, minor infections can be handled by the body's immune system, right?
Oh, yes.
So
Yes.
if one goes ahead to still give antibiotics for those minor infections, at a point the body would now um, um, would I say somewhat um, get too used to the antibiotics. Well, basically, the whole idea is that if the body can fight the antibiotics, there are minor infections, then there will be no need. The white blood cells are there to enable that fight to be successful if there are minor infections. Okay, of course. Um, we don't want a point where it will come to that. The body is now used to the antibiotics, and when the body actually needs them, it looks like um, the body is now resistant to the antibiotics that should help it fight the infection, the major infection. Okay, but what does the marking scheme say?
To reduce and prevent resistant strains of bacteria preventing. I
Mm-hmm,
mean, to reduce, prevent antibiotic resistance.
mm-hmm. Okay, okay. So yes, that's true. The body can even start resisting the antibiotics too. Okay, well, all right. Let's move on. It says figure 8 shows blood viewed using a microscope. Aha. It says a vaccine will stimulate the production of antibodies. Which part of the blood in figure 8 produces antibodies? Okay. So antibodies, I think that's a function of the white blood cell, right?
Yes.
So if we were to go with this graphical representation of the blood, I would think that letter A, would be pointing to white blood cells. But yeah, what does the marking scheme say for that?
It says B.
B for antibodies.
Yes.
Okay, B. Oh, but funny enough, B now seems to me seems to me as if they were red blood cells. Okay, so it says which part of the blood in figure 8 starts the clotting process? That would be white blood cells. So that would be red blood cells, sorry. So if B refers to white blood cells, then well Well, let's take a chance on it that A would be red blood cells. So are they good with that? That A is what starts the clotting process.
Yes.
Okay, okay, all right. That was a tricky one. Okay, this question is about cell division. Write the biological structure from the box in the correct order of size. Hmm. Okay, from the smallest to the largest. Okay, so chromosome. Chromosomes would be found in genes. Genes should be found in the nucleus. Nucleus would be found in the cell. Okay, so I think from smallest to the largest, I'll go with chromosome first, followed by gene, followed by nucleus, followed by cell. Is that correct?
Yes.
Okay. All right. Let's move on. It says figure 9 shows how a fertilized egg can produce specialized cells. Okay, embryo. Um, okay. So it says name the process A. A
Differentiation.
differentiation, yes, because that's at that point it's specializing into muscle cells, red blood cells, and the others. Yes, cell differentiation. Yes, I'll go with that. How many cell divisions are needed to form a 16 cell embryo from the original fertilized egg cell? Okay, so if we assume that each cell divides into two, you understand, and so on and so forth, how many times will that need to happen for us to have 16? That's just another way of asking that question. Did you get it?
Yes.
If you want to do a blank, uh, a quick mathematical assumption, do you think 8 will cut it? Do you think if we said 8, that would be correct?
Um. Yes.
Okay, let's see. So for my imagination now, a cell, I'm seeing a single cell, it divides into two. Each of those two divides into two again. I now have four. Okay, so how many has how many steps? Because it says how many cell divisions are needed to form a 16 cell embryo. Maybe I should even use. I'm looking for a writing material now because just using my imagination now, it's already it's looking confusing a bit. But let me see if I write it down, if I might be able to accurately tell what's going on. So let me see. Okay, so first cell division creates two. The next cell division will give us two out of those two. That's one, two, three. So another cell. So do you know what I'm doing? I'm breaking down each cell at each point. After three cell divisions, I already have one, two, three, four, five, six, seven, eight. Do you see that?
Yes.
So it's obvious that after four cell divisions, it's after four that I will get 16 because what I'm seeing here, I'm seeing one, two, three, four, five, six, seven, eight. And then the other one will now become one, two, three, four, five, six, seven, eight. That's where we have 16. So I'll go with four. So it's not actually mathematical reasoning that will get us that. It's understanding that each time there's a break, there's a cell division, one cell breaks into two. So each of those ones that broke into two will further break into two again. And you understand? And each of them would also break into two. That would rapidly increase the number of cells. Okay. So so from my drawing, it gave me eight 16 after four cell divisions. Do you understand?
Yes.
Okay. Do you want me to show you the diagram that I drew to achieve that 16?
I drew my own for the question.
Okay,
For the
okay, perfect. Okay, no problem. That's fine. Okay, let's move on then. Okay, it says in humans, the fertilized egg contains 23 pairs of chromosomes, correct? Um, how many chromosomes will there be in each of the embryo cells? It should be the same thing, right?
yes.
It should be the same thing. It doesn't change. It doesn't now produce um um another cell that has a different number of chromosomes, 23 pairs of chromosomes. It's the same. Okay. Figure 10 represents a cell cycle for a human embryonic cell. Describe one change in the cell that occurs during each of the stages of the cell cycle. Oh boy, I'm not too sure of this. I think we'll just have to go um with the marking scheme on this one. I think we will go with the marking scheme on this one. What does the marking scheme say about stage one, stage two,
Um,
and stage three?
Stage one, increase in numbers of subcellular structures.
Uh-huh.
Chromosomes double, duplicate, and replicate.
Okay.
Stage two, chromosomes pulled, one set of chromosomes pulled to each end of cell.
Mm-hmm.
Two nuclei form. Stage three, cytoplasm membrane divides. Two identical
Okay.
are formed.
Okay. 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 That's stage three, right?
Yes.
Okay. Okay. All right. Let's see. The next question seems to be having a graph somehow. It says cell division is important in the growth of multicellular organisms. Okay. It says this figure shows the mean height of boys and girls from birth to age 18 years. So what's the real question? I think we'll need to scroll down to see or scroll up to see the question below. It says compare the growth of boys with the growth of girls. Okay. Hmm. Okay, scroll up. Let's take a look at the graph. Let's see the things we can deduce from that graph. All right. First of all, I think uh, from 0 to 10, 11 years of age, it seems that girls, uh, the mean height of girls and boys seems to almost be the same. All right. We we start noticing a change at the age of 12, right? At the age of 12, it seems like the boys grow taller than the girls. And then after the age of 12, the girls, they just even out. They don't grow taller anymore. Okay? Do you agree with that?
Yes.
Alright. Okay. So let's see any, what's the next question on it after I turn? Okay, you want to scroll up or scroll down rather? I think we've done this, right? The comparison.
Yes.
Okay. It says give one way that cell division by mitosis is important in fully grown animals. Repair now. Repair of um body tissues. Yeah. I'll say repair of body tissues. Because if we say fully grown, that suggests that maybe something like height has completed and all that, so they don't need to grow again. But repair, repair, damage, and uh, yeah, repair of body tissues. So what does the marking scheme say?
Yes, repair of tissues or replacement of cells.
Mm-hmm. Okay, okay, all right. Replacement of cells. Okay, let's move on.
That's the last question.
That's the last. Oh, wow, excellent. And we did it exactly in 2 hours. Perfect. Okay, that's good. All right. So. Um, um, I'm not too sure now, but about when our next class is, but I think it's Monday.
Yes, I think it is also. Yes, Monday. 6:30 again.
Okay, okay. All right. So maybe we'll do chemistry or physics then, right?
My biology test is on Tuesday, and then my
Oh,
um
okay. So do you think we should do biology again on Monday? Yes,
Yes, please.
it's okay. All right, that's fine. Okay, that's good. So, till we meet on Monday. Enjoy the rest of your weekend and Sunday too.
Thank you too.